Bismillahirrahmanirrahim. Another video on some revision of this chapter, Transport in Humans, and I hope this is going to be helpful for you all. Now, looking at question number 1, figure 5.1 shows a capillary inside a tissue. Now, all the time, whenever you are doing such a question, read the question very carefully and explain to yourself. This is the blood flow. This is the lining of the capillary. So this is the capillary wall, and you can see there's got these cells in it, but it's got these spaces in between where something is going to come in or something is going to go out of here. So lining of the capillary, red blood cell, then we have R, Q, P, and then we have cells in the tissue, and we have this tissue fluid which is labeled, and then we have S, the arrow is going back into it, R, the arrow coming out of it. So take your time and look at it very carefully. And then let's look at the questions. Now the first question is, state how oxygen passes from the capillary into cell Q and describe the function of this gas in the cell. Into cell Q. Where is cell Q? Okay, this is cell Q. So why is oxygen going to move out and go into cell Q? So how oxygen passes? So how is a very simple answer, diffusion. So how is the diffusion? And then it says and describe the function. This is for 2 marks. So what is the function of the oxygen? Is aerobic respiration. Now you see, this is the way that you need to think. You did not do the whole sentence. You can always write in points. So if you just wrote like this, diffusion and aerobic respiration, used in aerobic respiration, you can say that as well. But so you just have to say that, and this got you your 1 mark for this and 1 mark for this, so 2 marks. Now it says part 2, name 2 substances required by cells other than oxygen that pass from the blood to the tissue fluid at R. So let's look at R. Here, what is going to name 2 substances? So when I say name 2 substances, then you need to be very clear what are you going to name. So what is going to be, what passes out is water. What passes out is glucose. You cannot say sugars, because sugars can be anything and they don't pass out. It's only glucose that passes out. And you could have said amino acids. Then you could have said ions. You could have said vitamins. And part 3, name 2 substances produced by cells that pass from the tissue fluid to the blood at S. So carbon dioxide. Carbon dioxide passes in the other direction. And you could have said water as well. You could have said urea. You could have said lactic acid. So all these were possible, and you could have even said hormones, because hormones produced by cells also enter the blood. Then coming to the B part of the question, with reference to figure 5.1 and describe and explain 2 ways in which capillaries are adapted to their function. Now as you can see here, there are 2 things. One is a describe and one is an explain. So you must think. So the describe first is it's got pores, or you can say holes, or you can say gaps in the capillary wall. This is what? This will be the describe. What is the explanation for this? Is that this is allows what? Allows the movement of small molecules between the blood and the tissue fluid. So the pores is the describe, and their function is allows the movement of small molecules between the blood and the tissue fluid. Then another describe is thin wall. And what is that going to help in short diffusion distance? So it doesn't have to cross a whole lot of distance, so it is a short diffusion distance. So this would be the describe and this would be the explain. Then you could have come and said something else. They are thin and narrow, so blood moves slowly. So description is they are thin or narrow, and the explanation would be blood moves along for exchange. Blood moves slowly for exchange. Then large number of capillaries. So there's a big capillary bed, and that provides a large surface area. So a lot of describe and explain. Number one was pores allows movement. Number two was thin walls, short diffusion distance. Number three was thin, narrow. Blood moves slowly. Next was large number of capillaries or capillary network, provides large surface area. Then it says tissue fluid drains into vessel P at T and eventually returns to the blood. Name the type of vessel labeled P. That's a lymph vessel. And then it says explain how fluid passes along these vessels, along the lymph vessel, because it doesn't have a heart, doesn't have a pump. So it is squeezed by muscles and there are valves which ensure one-way flow. So squeezed by muscles and valves ensure one-way flow. Then coming to question number two, the lymphatic system consists of thin-walled lymph vessels that drain tissue fluid from many organs of the body. It's lymph nodes that contain the cells of the immune system. The fluid in the lymph vessels is moved in a way similar to the movement of blood in the veins. Figure 4.1 shows part of the lymphatic system from the head, vena cava, direction of flow, ileum, from the legs. Suggest how lymph is moved in the lymph vessels. 2 marks. Basically, it's by muscles. We just did it in a previous question. This is more 0610 and valves prevent backflow. So muscles squeeze vessels and valves prevent the backflow. Then the B part of the question, after a meal rich in fatty food, the lymph leaving the ileum is full of fat droplets. Explain why there are fat droplets in the lymph vessel leaving the ileum. You see, the fat which you eat is digested to fatty acids and glycerol and is absorbed in the ileum to making the fat water soluble. So the fat enters the lacteals. Lacteals empty into the lymph vessels, and transport of fat in the lymph may reduce the risk of plaque in the arteries. So all the fat that you ate is not going to go into the hepatic portal vein, into the capillaries which join to form the hepatic portal vein. It's going to go into the lacteals. L for lacteals, L for lipids, L for lymphatics. So a very easy way to remember that. So fatty acids and glycerol absorbed in the ileum, fat enters the lacteals empty into the lymph vessels, and transport of fat in the lymph may reduce the risk of plaque formation in the arteries. Now coming to this part of the question, lymph flows through lymph nodes. Figure 4.2 shows the action of white blood cells in a lymph node when bacteria are present. So you can see the bacteria, they've shown you in the rod shape, and then they've shown you P. And what is P? P is, you can see mitosis, that one cell which is selected, dividing and forming many numbers of cells. And then they're producing antibodies, and the antibodies are going and locking on to the bacteria. And then the phagocyte comes, and here comes the bad guy, and that mops up all the bacteria. So it's like the police coming and rounding up all the criminals. So name the type of nuclear division shown in P. Of course, this is mitosis. Why? Because one cell is dividing into exactly genetically identical cells. So this would be, you can see this here, this is P here, this is P here, P here, this is P here divided into two. So this P had to be mitosis. Mitosis is the production of genetically identical cells, and that is, name the molecules labeled Q. What is that? Q is an antibody, because the lymphocytes are producing antibodies. So lymphocyte selection, then cloning, and then differentiation, and the antibody. Describe how the bacteria are destroyed by cell R. Cell R is the phagocyte. You can see the diagram here. This is cell R. So describe how bacteria are destroyed by cell R. Very simple. Phagocytosis by cell R. Antibodies are marking the bacteria. They're sort of attaching themselves onto the bacterial surface, so they are marking them. Then the bacteria are engulfed into a vacuole, and then enzymes are released, and the bacteria is digested and destroyed. So antibodies go and attach to the bacteria, and somehow they mark the bacteria, because now it will be easy for the phagocyte to come and identify them, and because they've been marked by the bacteria. So phagocytosis by cell R, then the bacteria is engulfed into a vacuole, and then enzymes are released and the bacteria is digested and destroyed. In question number 3, figure 2.1 shows a diagram of human skin in hot weather. Name structures A to D. What is A? A is the hair. What is B? B is the temperature receptor. Then C is the sweat gland. And D is the fat cell. The label, you can see the diagram. I haven't moved it upwards. So high hair, temperature receptor, sweat gland, and fat cell. Then coming to the B part of the question, describe how the structures A to D in the skin help to maintain a constant body temperature. You may refer to the structures by their letters. So here is A. Let's look at this. I'm going to explain it on this. So here is A. So what are we going to talk about? A is raises the hair and traps air. Air is a good insulator. Then what are we going to talk about? B. These temperature receptors detect change in temperature, and this generates an impulse. So this is the sensory neuron. So this generates an impulse in the sensory neuron, which goes to the central nervous system or the brain and the spinal cord. Then C is the sweat gland. What does the sweat gland do? It secretes or produces sweat, and this sweat evaporates from the surface of the skin. And that heat is lost from the body when the body takes the heat from the latent heat of vaporization. So heat from the body is taken when the sweat evaporates. It's not when the sweat is produced, nothing is going to happen. But when the sweat evaporates, it cools the body down. Now, what does D do? D is a fat cell, so it's an insulating layer. It's an insulator. So you can see this was a 4-mark question, and there were lots of marking scheme points. Number 1 was the hair, which raises the hair and traps the air. Air is a good insulator. Then temperature receptors, B, detect changes in temperature. Impulses are generated, and these impulses carry to the central nervous system, which is the brain and the spinal cord. Then C was the sweat gland. Sweat gland produces sweat and evaporates from the surface of the skin, and heat is lost from the body, or the blood is cooled. And D acts as an insulating layer. Part C, blood flows through the skin, changes in response to changes in the air temperature. State and explain what happens to the blood flow through the skin when the temperature of the surrounding becomes very cold. You enter this room is very cold. What is going to happen? Vasoconstriction, shunt vessel will open. Lesser blood will flow through the capillaries, blood diverted away from the skin. The fact that blood distributes heat. So if less blood is flowing in the skin capillaries, so then less heat will be lost by radiation and by convection to the air. And this part of the question was explain how the changes you have described in C1 reduce heat loss. And this you have to understand, this whole thing is for 5 marks. Now, actually blood distributes heat. And what will happen is, now that there is vasoconstriction, less heat will be lost by radiation, by convection, or by conduction to the air. The answer to this was, you see, blood distributes heat, less heat lost by radiation, less heat lost by convection, and less heat lost by convection and radiation by conduction to the air. Part D, the control of body temperature is an example of negative feedback. Describe how negative feedback is involved in the control of body temperature. You see, change in body temperature acts as a stimulus. Now, to keep the temperature constant, corrective action by the body takes place. So a change in skin temperature, why would change in skin temperature take place? Naturally, it depends on the environment. If this room is very hot, my skin is going to get very hot. If my room is very cold, the skin is going to become very cold. So changes in the skin temperature act as a stimulus. And to keep the temperature constant at 37, corrective actions are taken by the body. So when you enter this room is very hot, then there'll be vasodilation. If you enter this room and this room is very cold, then vasoconstriction to maintain the heat of the body and not get it lost to the environment. Question 4, figure 2.1 shows the blood supply for the liver of a mammal, vena cava, aorta, vena cava, aorta. Blood from organ X is carried to the liver by blood vessel Y. Name organ X. Now you could have said anything. Small intestine was correct. Esophagus was even correct. I don't agree with that, but then that's Cambridge mark schemes. So small intestine, ileum, duodenum. So any of these, even you could have allowed colon, rectum. Then blood vessel Y. What was blood vessel Y? Let's look at blood vessel Y. Let's look at it. Blood vessel Y. This is going in here from the intestine. So that would be, this would be, X would be the small intestine. From that is going to the liver. So blood vessel Y is hepatic portal vein. That's the only thing that connects the vein. So hepatic portal vein connects the small intestine to the liver. Hepatic, hepatitis is something to do with liver. Then figure 2.2 shows liver cells as seen with a light microscope. And then you have these diagram, you have P to Q, and then you label on figure 2.2 the structure A, B, and C. What is A? A is the nucleus. What is B? B is the cell membrane. And B is the cell membrane, C is the cytoplasm. So three for this label A, B, and C. So this was the 3 marks. Then the distance PQ. This distance PQ is 0.06 millimeter. Calculate the magnification of figure 2.2. Show your working. What we have to understand is we have to measure PQ, the scale, and this is about 120 millimeter. So what do you have to do is you have to divide, this is actually 0.06, so 0.0120 divided by 0.06. So this is about 2000 times. So this would get you your marks for this part of the magnification story. Cells absorb glucose and amino acids from the blood and help to regulate the concentrations of these substances in the blood. Explain how liver cells help to regulate the concentration of glucose in the blood in response to hormones from the pancreas in each of the following situations. So it's very simple. First of all, there are enzymes within the liver cells. Then there is a negative feedback, which is homeostasis. And then insulin produced by the pancreas, beta cells of the pancreas. So before that can be released and something happens, glucose enters or diffuses into the liver cells. Liver cells store glucose as glycogen, convert the glucose to glycogen, and so the blood glucose concentration is normalized. Then if the blood glucose concentration falls and you've been fasting for about 20 hours, then glucagon will have to be released. And this glucagon is released from the pancreas. The islets of the pancreas have the alpha cells which produce glucagon, and this is carried by the blood to the liver, and in the liver then this breaks down the glycogen to form glucose, and the glucose then goes out of the cells and enters the blood. So the first 2 points, enzymes in the liver cells, negative feedback, you could have written in any section, but for this section, blood glucose concentration is higher than normal, then you have to talk about insulin, glucose absorbed by the liver cells, and stores glucose as glycogen. But then under this heading, blood glucose concentration is lower than normal, then you have to talk about glucagon. The hormone glucagon, which is also produced by the islets of Langerhans, and this, of course, causes the liver cells to convert the glycogen to form glucose, and the glucose goes out of the cells and enters the blood. So glucagon, liver cells convert the glycogen, the stored glycogen to glucose, and the glucose goes out of the cells and enters the blood, and the blood glucose concentration normalizes. Describe what happens to amino acids inside the liver cells. Now, this is 3 marks. So, first of all, the amino acids are used to make new proteins, or we can say protein synthesis. Then the excess amino acids are broken down and deaminated. Then the removal of the amino group to form ammonia, converted to urea. And the remaining part is, of course, a carbohydrate. This is used to provide energy. And of course, it can be used used as a respiratory fuel. So what happens to the amino acids inside the liver cells? They are used to make proteins. So protein synthesis takes place. The excess amino acids cannot be stored. So the excess amino acids are broken down, and the removal of the amino group, and then they form ammonia, and then the ammonia combines with carbon dioxide to form urea, and the rest of the molecule is inspired to release energy. I've talked about this in in an older video of mine as well when I'm explaining this chapter. So this finishes this video on transport in humans. Many of you asked for a video on this so that we could do some questions on it, and wish you all the best of luck. Best of luck in the exams, just 2 days away. It's Sunday night and we have Monday, and then Tuesday morning you have the exam. So best of luck and all the very best from my side.